Hello friends, today I have to get idea about very interesting unit or topic. I have to say that is the cell structure and its types. My friends, we know that actually multicellular organisms are made up of more than two cells. Unicellular made up of a single cell. So you'll have to keep in mind we have been dealing with the plant physiology of a most of multicellular organisms. In the sense, the organism is composed of made up of a many cells. So it is necessary to find, we have to learn related with the difference between the cells, isn't it? Types of the cells, then structure of the cell, then parts of the cell, and again some cell organelles which are present in the cell, functions of those cell organelles. So that things today we have to learn. So in the beginning, my friends, we have to get idea about the difference between prokaryotic cell and eukaryotic cell. My friends, you have to keep in mind the living organism on this earth first time appeared as a prokaryotic cell, and then it evolved into a eukaryotic one. Then what is the difference? So again many more organisms are present on this earth planet are prokaryotic, and most of, we have to say that, organisms are the eukaryotic. So what is the difference between actually prokaryotic type of a cells and eukaryotic type of a cells? So friends, very minute size is there of a prokaryotic cell, but in case of eukaryotic cell, they are fairly large in shape. Before that one, I will divide the prokaryotic word into two parts. So in the first part, if you go, so there is a pro means primitive. Pro means primitive. Karyotic in the sense karyon, nucleus. Primitive nucleus. And in case of eukaryote, U means true. The cell shows presence of a true nucleus. And here in prokaryotic there is a primitive nucleus. So difference that we have to say. In case of prokaryotic, nuclear region not surrounded by nuclear membrane. So nucleus means true nucleus is not present in the prokaryotic cell. That nucleus we have to say as a nucleoid, which is not surrounded by plasma. So nucleoid is without plasma membrane structure. Whenever nucleoid is surrounded by the nuclear membrane, then that is the true nucleus. Whenever the nuclear material is surrounded by the nucleon, that we can find in case of a eukaryotic cell. Now in case of prokaryotic cell, genetic material, if you want to see, that is the single chromosome is present into that one. In eukaryotic cell, more than one chromosome present. My friends, already I have said to you, whenever the first organism appeared on this earth was a eukaryotic one, simple one. Later on the evolution takes place and the system, cell system and plant system, tissue system, and we have to say that the organ system and again organism system becomes a little bit complicated, isn't it? So that we can see here the prokaryotic cell shows presence of a single chromosome, but in eukaryotic cell more than one chromosome is present. Now in case of prokaryotic cell, nucleolus is absent, nuclear membrane is absent, and again nucleolus is absent. In case of eukaryotic cell, nucleolus is present. My friends, another thing is that in prokaryotic cell, membrane bound cell organelles are absent. So without membrane we can find the cell organelles in prokaryotes, but in membrane bound, in case of the eukaryotic cell, membrane bound cell organelles are present. In case of prokaryotic cell, cell division is by fission or budding. No mitosis takes place. No mitosis takes place. But in case of eukaryotic cell, mitosis and or meiosis, we have to say that cell division occurs. Once again, I will give you an idea about the difference between the prokaryotic cell and eukaryotic, because these things help us to understand the physiology of a plant. Structures of the organs, structure of the cell, type of the cell should be known to you very, very clearly. Then it is possible. Once again, I will tell about the prokaryotic cell, difference between the prokaryotic cell and eukaryotic cell. Prokaryotic cells are minute in size. Eukaryotic cells are fairly large in size. In case of nucleus in prokaryotic cell, nuclear region, that is nucleoid, not surrounded by the nuclear membrane. But in case of eukaryotic cell, nuclear material surrounded by a nuclear membrane. In case of prokaryotic cell, single chromosome is present in that cell. But in eukaryotic cell, more than one chromosome present. In prokaryotic cell, nucleolus absent. In case of eukaryotic cell, nucleolus present. In case of prokaryotic cell, the cell organelles are without the cell membrane. Sorry, without the membrane. But in eukaryotic cell, each and every cell organelle is surrounded by, bound by the membrane. In prokaryotic, fission or budding type of a cell divisions occurs, means no mitosis. But in case of eukaryotic cells, cell divisions, that mitosis and meiosis occurs. Now, advantages of each kind of cell architecture that we have to study. Advantages of each kind of a cell architecture. In case of prokaryotic, simple and easy to grow. Prokaryotic cells, simple and easy to grow. Eukaryotes can specialize. Fast reproduction is possible in case of a prokaryotes. In eukaryotes, multicellularity is there. In prokaryotes, all the cells are same size, but in eukaryotes, it can build a large, complicated bodies. My friends, once again, the difference is here in case of eukaryotes and prokaryotes is there. 10 to 100 micrometer diameter of the eukaryotic cell, which is a bigger one. In case of prokaryote, 0.2 to 2 micrometer diameter, is smaller in size. As already we have seen, membrane bound nucleus and nucleolus cell organelles we can find in eukaryotes. But in case of prokaryotes, we cannot found, or we can find unbound nucleoid, unbound nucleus and unbound cell organelles are there. In case of eukaryotes, mitosis, meiosis takes place. In case of prokaryote, no mitosis, meiosis, but cell undergoes the division by budding or fission. Cell wall simple when present. Cytoskeleton, big ribosomes occurs in a eukaryotes. In case of the prokaryotes, small ribosome and no cytoskeleton is there. Some things are common in between eukaryote and prokaryote, that is flagella. In case of plasma membrane, cell division takes place, cytoplasm is present, and ribosomes and chromosomes. As we have seen, simple chromosome, single chromosome present in a prokaryotes, but we have to say that the many chromosomes present in eukaryotes. Again, my friends, when you want to understand the plant physiology of unicellular or multicellular organism, you could have thorough idea about the cell of that plant, because cell is responsible for conducting many more plant physiological activities in the plant. So here plant cell, we have to get idea about that one. The plant cell structure is about you. It shows presence of a cell wall, which is a dead one, isn't it? Now that cell wall is followed by plasma membrane. First of all, cell wall we have seen, then that is followed by plasma membrane. Then in plant cell we can find Golgi apparatus. This structure is a Golgi apparatus. Then Golgi vesicles are also there. Small size Golgi vesicles are there. Also, the plant cell shows presence of a chloroplast, larger size vacuole. This vacuole is surrounded by vacuole membrane. Again, we can find raffied crystals in vacuole. Other crystals are also in the vacuole. Mitochondria, which is we have to say that the energy house. Then all these cell organelles, most of we can find that are in a cytoplasm. Again, large central vacuole, already I have given you idea. Then nucleus is there, isn't it? This nucleus shows a nuclear membrane and extension of a rough endoplasmic reticulum. Here we can find, and again smooth endoplasmic reticulums are here, isn't it? Chloroplast is the most important part of a plant cell in case of green plant. So this is a structure. You have to sketch this structure, label its structure 3-4 times by viewing the structure, viewing this slide. But again you will have to try to sketch the diagram and label the parts of this cell structure without looking into the book or slide. Now my friends, this is we have to say the ultra structure of a plant cell that you will be able to see. We have seen many more cell organelles: vacuole, nucleus, endoplasmic reticulum, Golgi complex, smooth endoplasmic reticulum. Then in nucleus also there is a nucleolus, nuclear envelope, nuclear pore. Most important things, that is of a cytoskeleton. That is so this is ultra structure also you will have to sketch, study properly. So you will come to know that how the cell works and how the parts of the we have to say that the cell organelles or each part of the cell is involved. Part of the cell involves into the physiological activity of the plant. Now, animal cell and plant cell, there is a difference into that one. That also we should know which things are present in the plant cell. Cell wall is present, chloroplast is present, large vacuole is present. These three things are absent in a animal cell. Which one? Cell wall, chloroplast and large vacuole. But centriole absent in the plant cell. Remaining, that is a plasma membrane, endoplasmic reticulum, mitochondria, nucleus, Golgi apparatus. All these are the common one. So by looking into this one also you will come to know that. Again I will tell you, as a plant, I have to say that the teacher of a botany, plant cell structure, which things are, means the difference between the animal cell and plant cell we have learned right now. In plant cell, the extra part which are those, that is the cell wall, chloroplast and large vacuoles which are absent in the animal cell. But centriole is the part we have to say that absent in a plant cell. Other things are common. So here diagrammatically you will be able to see which are the common things which are present into that one and which are absent, which are into that one. That also will be seeing this will understood. So this is the difference between animal cell and plant cell. So again, how are the plant and animal cells are different, that are shown here to you. As already we know that vacuole, I will show to you this vacuole which is not present in the animal cell. Most important. Again, in case of the chloroplast, this chloroplast is also not present into this cell. Another cell is cell wall. Keep in mind this cell wall structure which is not present in this animal cell. So you have to keep in mind these three structures are absent in a animal cell but present in a plant cell. But centrioles, very interesting thing is that, which are present in a animal cell, the structure is shown here. Centrioles which are shown here, which are absent in the plant cell. So this is the difference between plant cell, animal cell. So right now in this lecture we have got idea about difference between animal cell, plant cell. Again we have studied in the beginning of this lecture that is difference between the eukaryotic cell and prokaryotic cell. So these things known to you very well, because this is the actually foundation of learning of the plant physiology or any plant botany branch. Again, sketch all these diagrams, my friends. I have given you homework to you. Everybody will have to do this so as to understand the things very well. Again, this is the table. This table also gives us idea about the difference between the plant cell and animal cell. We have to keep in mind here cell membrane is present in this plant cell. This is a animal cell. So cell membrane is present, nucleus is present, nucleolus is present, ribosome is present, endoplasmic reticulum, Golgi cell is present, centriole. This is actually plant and this is the animal, my friends. Centriole is not present in the plant cell, isn't it? Centriole is not present in the plant cell. In case of cell wall is absent in animal cell but it is present in plant cell. Again, chloroplast is not present in this animal cell but it is present in the plant cell. Again, big vacuole which is present in the plant cell but which is absent in the animal. So this is we have to say that again table form in word structure in all. I have to say that things have been used here to make you clear the structures, difference between the structures of animal cell, plant cell in this lecture. Now again, exact structure of a plant cell we have to study, and then we will again come to know that what is the role of that part in the plant cell. First we have to take into consideration that is of a cell wall is there. Cell wall is a very strong characteristics of the cell wall. Cell wall is made up of a cellulose, pectin, lignin, hemicellulose, cutin, suberin. So these actually deposits helps cell wall to protect the inner material of a cell. What are the functions? So I had definitely protective, protect cell from the rupturing, protect the cell from mechanical injury, glued to the other cells next door, provide a rigidity, shape, exact shape of the cell remain as it is in the organisms as long as the organism is in a living condition. Support the shape, helps in transport. Cell wall helps in a transport. You have to keep in mind cell wall helps in a transport. This is a cell wall. Okay. Now cell membrane. Cell wall is followed by cell membrane, my friends, next to cell wall. Now it surrounds the cytoplasm. Plasma membrane surrounds to the cytoplasm found in the cell. What are the functions? Shape, gives a proper shape, protects the inner material. Cellular permeability means we have to say that the material can passes through that one. Permeability is there. Reabsorption is excretion, secretion, formation of various cell organelle is also done by the plasma membrane of the plant cell. Again, it provides protein, serves as an enzymes, selectively permeable means it never allows the entry of each and everything inside the cell or each and everything outside the cell, means exoosmosis and endosmosis at the time of that one. It acts as a receptor. Unicellular helps in the formation of a pseudopodia. Actually this is You have to only get idea about the plant cell itself. Now my friends, structure, ultra structure of a plasma membrane, because this structure will help you to understand actually the how the things are going to happen. So here you see glycoproteins are there, external membrane surface. So it is a fluid mosaic model. Fluid mosaic मॉडेल इज मोझॅक म्हणजे आपण मोझॅक टाईल्स बघतो, सिमिलर बाय इथं टाईल्स लावले ते मोझॅक टाईल्स दिसत आहेत आणि इथं मध्ये प्रोट्रूड आउट आलेला जो स्ट्रक्चर आहे दॅट इज ऑफ ए प्रोटीन्स and these glucose molecules are joined to the protein, that's why it is known as a glycoprotein. This is a polar end is there, and below that one, so protein, this plasma membrane are made up of a two types of a molecules, that is lipoproteinous in nature, made up of a lipids and proteins, made up of a lipids and proteins. So you have to keep in mind lipoproteinous in nature, lipoproteinous in nature. So this is the structure. That structure helps to understand the things. Now some proteins acts as a channels, entry of some substances, and we have to say that there is a coming out some structures through these channels takes place. So these are the known as a protein channels. हे जे स्ट्रक्चर आहे मी बऱ्यापैकी रेड करतो, दॅट इज अ प्रोटीन चॅनल्स आर देअर, प्रोट्रूड आउट, हे जे बाहेर आलेले आहेत ते प्रोटीन्स आहेत, इजंट इट, एम्बेडेड इन टू द लिपिड मोलिक्युल्स, एम्बेडेड इन टू द लिपोप्रोटीनस इन स्ट्रक्चर. सो पोलार एंड्स आर आउटसाइड, दोन्ही पोलार एंड आहे, फॉस्फोलिपिड बाय लेयर, फॉस्फोलिपिड बाय लेयर. सो हा एक लेयर आहे, दिस वन इज लेयर, फॉस्फोलिपिड बाय लेयर, एक लेयर आणि हा दुसरा लेयर, एकावरती एक, अँड द प्रोटीन्स आर एम्बेडेड इन टू दॅट वन. सो पोलार एंड्स विच आर आउटसाइड अँड नॉन पोलार एंड्स दॅट इज इनसाइड, वी विल बी फाइंड आउट टू द lipid molecules. So this is the little bit about the structure of the plasma membrane. Again, polar end, phospholipids are there, head, you have to keep in mind head, and this is a tail region, hydrophobic in nature. These tail regions are hydrophobic in nature, in the sense they are water repellent, hydrophobic, and that are non-polar end. But polar heads are hydrophilic in nature, they love water molecules. So this difference you will be able to understand very well. These are the phospholipid molecules. So phospholipid molecules, polar ends are this circular, this is polar end, things are there. Now again in detail the structure of a plasma membrane we will find here. So what happens actually, this is internal cytoplasmic region, and that cytoplasm is surrounded by this plasma membrane. This is a total plasma membrane is this one, isn't it? So in this plasma membrane we will be able to find out the protein molecules. These are the protein molecules which are embedded. These are the channel proteins plays a role as a, and these protrude out. So fluid mosaic model we will be able to find out. This is the outer side polar end, again this is the inner side polar end, in between hydrophobic in nature, non-polar end of the phospholipids are there. So these glucoproteins, so what happens, a glucose chain have been attached to the proteins. So glucoproteins are there, glycoproteins, here word is written, transmembrane structures are there. So carbohydrate molecules are attached to the proteins, that's why that is a glycoprotein, transmembrane. So it is a, from one end to another end we will be able to find out these protein structures. Then this is a protein, peripheral protein, because they are on one side of that's a peripheral in nature. So this is again exact structure of a plasma membrane. Again you see fluid mosaic model, each and everything you will have to always, all the diagrams you have to sketch, all the diagrams you have to then label properly, try to understand the structure, what is hydrophilic in regions, what is hydrophobic in nature, that you will have to study very well. Again same structure is there, how the things are going to happen. Already I have given you idea about the structure of the plasma membrane. Again this is different type of a membrane transport that we have to study, my friends here. So here hydrophilic polar end head, hydrophilic, these are the polar ends all of a lipid molecule. Now simple diffusion process ना काय झाले, आउटर वरून इनर साइड साइटोप्लाझम मध्ये मटेरियल आलं. Simple diffusion process. Facilitated diffusion in the sense facility has been provided by the protein channel. हे protein channel आहे आणि त्याच्या थ्रू आलेलं आहे, facilitated. Here we will be able to say that again molecule to be transported inside. But for that one active transport, because energy is used here. ATP molecules have been used here, isn't it? And this here the molecule transferred inside the cell with the help of a protein channels. Membrane transporter proteins are there. This portion, blue portion, is a hydrophobic non-polar and fatty acids tails of a phospholipid are there. Now extracellular space, this one all. Cell membrane, total structure is cell membrane is there. Now substances leaving the cell कुठले substances सोडून देते, secretion, nitrogenous waste, oxygen, carbon dioxide, excess water. The substances entering the cell आत कुठले येतात, the nutrients, ionic salts, oxygen, carbon dioxide, water that comes inside the cell. Now my friends, here this slide you will be able to see, which gives us idea about a types of solution, that is of a isotonic solution, hypotonic solution, hypertonic solution. My friends will have to keep in mind. So isotonic solution means such a type of a solutions actually not available in the anywhere in the world. But what happens actually, because of their concentration some differences are there. Means what I am going to tell you, suppose we are going to say that Balu is a heighted fellow. आपण हायटेड फेलो, त्याला हायटेड कधी म्हणू शकतो, तर बाकीचे त्याचे फ्रेंड्स हे त्याच्यापेक्षा ड्वार्फ असले पाहिजेत, इजंट इट. सो कंपॅरिझन आलं तरच आपण सांगू शकतो की मोठा का बारका. सिमिलर वे सोल्युशन्स हायपरटोनिक, हायपोटोनिक आणि आयसोटोनिक असं आपण कुठल्या सोल्युशनला म्हणू शकत नाही, पण त्यांचं कंपॅरिझन केलं ऑन द बेसिस ऑफ ए देअर कॉन्सन्ट्रेशन, देन ओनली इट इज पॉसिबल टू से दॅट हायपरटोनिक, हायपोटोनिक अँड आयसोटोनिक सोल्युशन. पहिलं बघ आयसोटोनिक, आयसो मीन्स सिमिलर, मीन्स इनसाइड द सेल. ही जे रेड दिसते आपल्याला त्या सेल स्ट्रक्चर दाखवलेलं आहे, आणि ह्या सेलमधल्या असलेल्या सोल्युशनचं कॉन्सन्ट्रेशन, त्याच्यामध्ये असलेले सोल्यूटचा नंबर लक्षात येते ना, हा बाहेरच्या असलेल्या सॉल्व्हंटच्या नंबर एवढा असेल, येतं आहे का लक्षात. म्हणून त्याला म्हणायचं आउटर सोल्युशन इज अ अँड इनर सोल्युशन इज अ आयसोटोनिक वन. No the soling of a solvent will be takes place and no shrinkage takes place. लक्षात येते का ना, कारण ह्याचं आणि ह्याच्यामध्ये असलेले सोल्यूटचं कॉन्सन्ट्रेशन आत आणि बाहेर सारखंच आहे. आता हायपोटोनिक्स, कुठल्या ह्याला हायपोटोनिक म्हटलं आहे, जे सोल्युशन सेलच्या बाहेर आहे ह्याला हायपोटोनिक सोल्युशन म्हटलं आहे. हायपोटोनिक वाय, बिकॉज सोल्यूट जे आहेत बघा इथं विरळ दाखवलेले आहेत, सोल्यूट्स जे गोल दाखवलेत बारके बारके सोल्यूट, पण ह्याच्यात जर त्या सोल्यूटच्या आपण आत बघितलं तर त्या सोल्यूटचं कॉन्सन्ट्रेशन हे जास्त आहे, म्हणून बाहेरच्याला हायपोटोनिक म्हटलं आहे. मग आतलं सोल्युशन हे हायपरटोनिक असणार. मग हायपरटोनिक असल्यामुळं सॉल्वंट बाहेरून आत जातो आहे, म्हणून हे फुगलेलं आहे, मोठं दाखवलेलं आहे. आर यू गेटिंग? ॲज कम्पेअर टू दिस वन मोठं दाखवलं का, तर ह्याच्यामध्ये सॉल्वंट गेलं. आर यू गेटिंग? पण हे सॉल्वंट किती जाईल, अनलेस ना अंटिल देर इज अ बॅलन्स इन बिटवीन द आउटर कॉन्सन्ट्रेशन सोल्युशन अँड इनर कॉन्सन्ट्रेशन सोल्युशन गेट इक्वल और गेट आयसोटोनिक. सिमिलर वे इथं आता बघा हायपरटोनिक. आता ह्याला बाहेरच्या सोल्युशनला हायपरटोनिक का म्हटलं, तर बिकॉज सोल्यूट बघा, सोल्यूटचा नंबर बघा दिसतोय काढलेला, दॅट इज अ हायर ॲज कम्पेअर टू द इनर, आणि त्याच्यामुळं काय झालं, आतलं पाणी हे बाहेर आले, म्हणून हे श्रिंकेज झालं. म्हणून दिस इज ए श्रिंकेज टेक्स प्लेस, बिकम्स ए स्मॉलर इन साईज. आर यू गेटिंग? हायपरटोनिक. मग बाहेरच्या सोल्युशन आपण हायपरटोनिक का म्हटलं तर सॉल्वंट त्याच्याकडे येत होतं. ज्याच्याकडं सॉल्वंट जातं आहे ते सोल्युशन हायपरटोनिक. हे लक्षात ठेवा. पुन्हा एकदा मी सांगेन सिम्पल थिंग्स. आजपर्यंत तुम्हाला लक्षात ठेवायला अवघड जायची गरजच नाही. साखरेकडं पाणी पळतं आहे. साखरेकडं जिथं साखर जास्त तिकडं पाणी पळणार. किती पळेल? आतला आणि बाहेरच्या याचं इक्वल कॉन्सन्ट्रेशन होईपर्यंत, सोल्यूटचा नंबर एकसारखा होईपर्यंत. लक्षात येते ना, सॉल्वंटमध्ये त्या सोल्यूटचा नंबर सेम होईपर्यंत. ओके. हायपरटोनिक, हायपोटोनिक आणि आयसोटोनिक. तर डायरेक्शन ऑफ ए ऑस्मोटिक वॉटर मुवमेंट कुठं होते? आयसोटोनिकमध्ये कुठेही मुवमेंट होत नाही, दोन्हीकडेही सारखं कॉन्सन्ट्रेशन आहे. हायपोटोनिकमध्ये काय होतं आहे? सेलमध्ये हायपरटोनिक कंडिशन असल्यामुळं तिकडं सॉल्वंटची मुवमेंट होते, एंडोऑस्मॉसिस. हायपरटोनिकच्या बाबतीमध्ये म्हणजे हायपरटोनिक बाहेरचं सोल्युशन आहे म्हणून एक्झोऑस्मॉसिस, सेलमधलं पाणी बाहेर येत आहे. ओके. अगेन सेम स्लाइड इज देअर. In case of mitochondria, that you have learned very well, structure of the mitochondria and function, that is of a energy production. Double membrane bound, outer membrane thick, freely permeable, inner membrane selectively permeable, infolded, called as a cristae. That is learned by you. Then also once again I am going to revise the things. F1 particles or oxysomes on a cristae. Oxysomes having a head, stalk, base pieces are oxysomes, means this structure. Okay, so this is all about the mitochondria. You will have to go through this, sketch the diagrams and try to understand the parts of the mitochondria. Again in detail here given the, already I have told you mitochondria are responsible for the breakdown of a sugar molecule to release the ATP, adenine triphosphate, which is used to transport the energy within the cell for the metabolism. These are the parts, the head, stalk of these F1 particles are there. Am I right? In details, chloroplast, because we see, my friends, we have to understand first of all the structure parts of the cell organelles, because they are playing an important role in the physiology of a plant itself. So this is all about the plant cell, sketch diagrams, try to understand the things very well. Now in case of the nucleus also, already we have studied in a detail about the nucleus. Then nucleolus is present inside the ribosomes. Function of the ribosomes you will have to again understand. Build the proteins from the amino acids in the cytoplasm, may be free floating or may be attached to the endoplasmic reticulum. Endoplasmic reticulum made up of a RNA. Endoplasmic reticulums may be smooth, build cell lipid and carbohydrates, may be rough, that is a rough endoplasmic reticulums are there. This stores the protein made by attached to the ribosome. Golgi complex takes a sacs of a raw material from the endoplasm and sends out the sacs containing the finished cell products. Lysosomes are there. Sacs filled with the digestive system of these lysosomes. So digest the worn out cell parts, digest the food absorbed by the cell taken place by the lysosomes itself. Centriole sphere of a bundled tube that we have. Now cytoskeleton, that is most important to make the cell rigid in structure, made up of a microtubules found throughout the cytoplasm, gives shape to the cell and moves the organelles around inside. So cytoskeleton helps to that. Then in case of the vacuole, huge water filled structure is there. Huge water filled sacs, that is a vacuole is there. Keep cell pressurized, stores a starch. Again these structures also will help you to understand about the cells and cell organelles, how the microfilaments and other structures helps to give a specific shape with the help of a cytoskeleton. In case of endoplasmic reticulum, the smooth endoplasmic reticulums are there that are without any type of a substances present into that one. But in case of other, we can find the deposition, so it becomes a rough structure. Then centrosomes are there. This is all about the Golgi apparatus. Fusion from secondary lysosome, primary lysosome. So that cell organelles are produced. chloroplast.